Okay. Welcome back, everybody, for our new webinar. For those of you that are watching as a recording, I am so excited about today. I feel like meeting Julia was my Cure PDE highlight of 2024, actually. So we met at NORD, at the NORD conference, where we just happened to sit next to each other. And then she told me about her journey with her two boys and the path that she has essentially taken, and she'll obviously give you a better description, but the path that she's taken of taking therapeutic development into her own hands. And I guess it shifted my whole perspective on the way that we as a community can work together to shape kind of our own future and destiny when it comes to what's available for our PDE kiddos. So I don't expect that our journeys are going to mirror Julia's exactly. And of course, all of our kids and all of our communities are different, but I just wanted her to very graciously share her story with us. And she actually just came out with a book about it, which is amazing. So hopefully she'll share some of that information with us. But I just really want you guys to pick her brain and listen to the story and see where the takeaways are for us as a community, thinking about what we could actually accomplish just sort of with our own, I don't know how you would say it, willpower and passion, I guess. So I'll hand it over to Julia and yeah, take it away.
Well, thank you. Thank you, everybody, and nice to meet everybody. Well, yes, just like Anna said, that we would just sit next to each other at NORD and just start talking. I mean, I guess whenever you are at those rare disease conferences, you just talk to everybody and anybody because everybody has story to share. And so we start talking. And that's why it's end up in a meeting today. And hopefully, you will be able to do something with that information. It is hard, but it's not impossible. You have a pretty big community. I can see that with the people online, and I'm pretty sure there will be several people that cannot kind of join and will listen offline. So that's for the ultra rare diseases like yours and like mine. It's actually considered to be a pretty big community, and you can do something about that if you put your heads together and kind of push through that. I actually put together a little presentation. That's what I was going to share through my computer.
Letting you share?
Yep. It's kind of slides from everywhere, just what we've been doing, and a few slides how it can be approached and what you need to look at before starting the work.
We see it. Yep.
Let me see if I can put it in the slide mode. Yeah. And so in the end, so I started that foundation that's I call Rare Trade Hope Fund. I started the foundation 12 years ago. And originally, I wanted to be just an disease advocacy foundation, just like everybody wanted. But literally what happened with the ultra rare diseases, slowly the foundation is converted. What I mentioned here is to nonprofit biotech, because majority of the stuff that needs to be done, there is not a single entity out there so far that I know, the ones that can handle all of the aspects of the drug development. There are entities, drug development for ultra rare diseases. There is a lot of entities, pharmaceutical companies that will do drug development. But the goal of the pharmaceutical company is to develop something that they can, after extract profit from them. That's in their bylaws. And that's how the shareholders get finally return on their investments. And that's kind of a knowledgeable cause, noble cause. But with the ultra-rare diseases, the probability of getting profits is very, very slim, close to none. And that's one of the reasons is there is entities, commercial entities would not be, it's very difficult to find somebody interested in developing something like that. So it's, of course, it started with my kids. That's my kids. Daniel is the oldest boy, and then Alexander came along about 5 years later. By the time Alexander was born, I knew that something is not fully kind of normal with Daniel. He was not failing preschool, but he was not an average child, which between me and my husband, having college degrees, I was hoping that he would be above average. But he was like one of the, because we spent a lot of time working with him, but he was close to failing the kindergarten. Back then, I tried to go, so that was a long time ago. So my oldest one, well, there is no but the oldest one is actually just turned 30 this year. And the youngest will be 25 in a few weeks. I never could figure out what's wrong. So the kids were both diagnosed eventually with autism, ADHD, and was getting support at school, but nothing kind of could help propel their development. Eventually, the older ones start repeating grades, I think like maybe first grade and then fourth grade. And it doesn't matter what we try to do. We try to do extra curriculum activities, meaning that extra training. It didn't work. I tried to go to the doctors to try to get diagnosis. In our case, the diagnosis was a long time it took. Eventually, when we got diagnosis, Daniel was 18. By the time, majority of the doctors will actually give up giving diagnosis. And if you remember, what happened is gene sequencing tests start coming out online. And what I did is actually I bought the gene sequencing kits like a couple of years before that to everybody in the family. But of course, like the regular data will not give you the information, at least at that time, about those ultra rare diseases. So couple of years in Macon, when I went to taking semester at university for the genetics, I actually downloaded the raw data from the gene sequencing files for everybody in the family and wrote the program, and actually end up figuring out that the kids have this rare disease, and the rare disease called aspartyl glucosamine. From that kind of name, it's actually tells you what's the problem. So it is the enzyme, so the gene that's not working, that missing the enzyme that breaks down certain type of the glucose that's connected to the aspartyls, and the left that connected through aspartyls, and the leftover of those unbroken sugars are passing through the urine. So this is the resulting in the non-functional gene and the gene called AGA gene. What is the problem and the science of the disease? It's the first one is the speech and language delay, speech problems, language delay, and developmental delay and intellectual disability. In between those two, all of the kids are diagnosed with autism. Majority of the kids have skeletal abnormalities. About 50% of the kids have seizures or end up developing seizures. And resulting in a premature death. Somewhere between, well, the reason why it's put, so once we start working on that, the premature death was known to have in people between 25 to 35, which is still the case in less developed countries. And this is majority of that happening actually due to those particular enzymes is being very prevalent in their immune system. So once the young adult, the person with AGU gets some kind of issues with the infections, that's usually the results of the cause of death. So in the more developed countries, with the better care, the life extension can be extended by around 10 years. But that's it. And that's what I was actually end up with the diagnosis, and that was in 2012. So majority of the work from 2012 to now was trying to figure out how to develop the treatment. The reason why we choose gene therapy, there is several things that can be done. There is a few things that can be worked on if a child, a person have a genetic disease. So it is some type of way to work on, let's say, genetic manipulation or genetic work. And I'm pretty sure Anna knows it very well, and she probably can answer all of your questions of that. Before we started the research in gene therapy, I had several meetings. So the kids were diagnosed. They finally got the final diagnosis, because once I got the results from my kind of gene therapy assessment, genetic assessment, I had to go back to the doctors and specifically ask them to test on this particular gene. So we got a final medical diagnosis in May of 2012. So the next 6 months, I spent time to try to figure out what we're going to do, because that diagnosis from the doctor came with the results. With the result is, I'm sorry, but there is nothing we can do for your child now. And what we did is, so my job in my previous life, and at that time I was still having a full-time job, is actually doing the project and company developments at the early stages. So we have some problem. That was my job. Was I had a problem and I had to figure out what to do with that project to make it viable. How to put the project together, how to find money, how to put people together. So that was literally my job. So that's what I did for the 6 months. What I did is I kind of almost knew that I couldn't put all eggs in one basket and try to figure out that I need to go with one thing. So I put a few people together, asking them one question: what approaches and what work do we need to do in what avenues to be able to figure out the treatment for my kids and for everybody with AGU. And we had we were offered several results, so several options. And out of several options, we did the assessment. It's called risk assessment. Essentially to, in our particular case, we were trying to figure out what will it take to bring that therapy to the, let's say, to the clinical trials and maybe to the approval eventually. How long it will take, that was very important for me. And then whether my kids, so because the length of will also relates to whether my kids will be eligible or not. Because if the timeline is way too long, the kids might not be my kids might not be eligible to that, which I will still would be work on that, but with a kind of different mindset a little bit. But also, how will it will that particular treatment will be just for my kids or also for everybody in the community? And as a result of that, what we found out that the gene therapy would probably will be suitable for every single person that has the mutation in the AGA gene, and because the gene replacement therapy would be able to kind of work on not only mutations, but also like something that's called like deletions, duplications, or all kinds of different genetic problems with the gene. On the other side, I kind of told myself that we needed to do some other gene therapy, because when looking at that, and you can see the timeline that it took to get to the clinical trial, and we haven't started it yet, so it will start this year sometimes. I also worked on something at the same time, parallel, that will be able to kind of sustain my kids or help them deal with their problems right now. And the second treatment was something that's called chaperone therapy or small molecule therapy. And I think you're all familiar with that. I think your disease is one of the ones that actually have that, correct, Anna?
I don't know if that's what we call it. I don't know if Curtis is on. I mean, we have a way basically to keep the kids functioning with B vitamin supplementation. Yeah.
Yeah. So essentially so there was none like that for AGU, but literally that's what we worked on. And it's also one of the supplementation. It's also in a like large amounts, and this supplementation varies a little bit dependent on mutation.
Yeah.
So, but it's also extremely large amounts, and the child has to take it every day, like 2 or 3 times a day.
Yeah.
So it's very similar to what you're doing.
Exactly. And that's actually one thing that I found really amazing is the idea that we don't, I guess, like that there could be multiple tracks, right? Like we can work on one while we work on the other. Obviously, we have, we have dietary guidelines for PDE. And this is definitely not saying to abandon any of that. And there's
Yep.
other trials that Curtis has talked about with, substrate inhibitors, et cetera. None of those things need to be sort of antagonistic to each other. And that's one of the things that I loved about your story too, is that you worked on both in parallel, looking forward to downstream gene therapy, basically.
Yeah, so one of the things that, and that's one of the things that why I really like encourage almost everybody whom I talk to, that you cannot literally do only gene therapy or only something. You have to work on bunch of things in parallel. If you look at the timeline and any timeline for any other disease, ultra rare disease, that development timeline, the parents that started it, if it isn't works for 10 years, their child is not eligible because the disease progression is so kind of dramatic. The child, by the time the clinical trials come on board, the child is not eligible for it. But because of that small molecule therapy that my kid's been taking since 2015, since like probably end of 14, that was developed pretty quick, like in about 2 years. My kids are still eligible, so we just went to neurologist to do the assessment. My kids are still eligible for the gene therapy. And eligibility for gene therapy, what I mean is if the disease, so in a treatment, and that's probably like you have over here, like Anna and I think Taisi, it's a lot better than me to explain that the disease, genetic disease progression, kind of the way how I looked at that, to put it in more simple terms, is there is two parts to it. Once it's something is not functioning, and then you can reverse it. But then also, once the disease is progressing, what it means is that non-functioning is causes some kind of either cell death or deterioration.
There's a
or
reversible piece and a non-reversible.
Yeah, exactly.
Yeah.
So, and that's what, so when the cells die, whenever your limb is getting amputated, that's just dramatic. Whatever. There is nothing you can do with the so that's not the part that we're working on. We're still working on the reversible part, and that's why you have to keep the system alive till that kind of fix will come to life.
Julia, what was the small molecule you discovered? I think you had told me that it also started with something simple, right? The initial,
Yep.
the very first small molecule was what? I forget the story.
The very small molecule. I mean, the very first time, that's when we started that. It actually was, and everything is published. So the very first one was glycine. It's
Lysine,
one
right?
of the amino acids, and
Yeah.
it is the smallest amino acid. And what it does, in my kid specific mutation and in like several other mutations, if it's present all the time, it allows the folding of the protein properly.
Oh, okay. Yeah.
So that's
Yeah.
what happens. But then we did some work, and that glycine kind of changed to betaine, which is it's kind of, it has the methane. It's more chemical. We're talking more chemistry here, but it's betaine is very similar to glycine with just few methane ends to it. And then it was actually adjusted specifically for my kid's mutation, because it was looked at a bunch of different, betaine was looked at a bunch of different mutations and they find out. So for my kids, there's another acid, just because of the polarity of their mutation. One other amino acids is a little bit better than glycine. So it's kind of developed throughout the years.
Yeah, I think that's so
So,
cool. Yeah, go ahead,
yeah.
continue.
But this development timeline, what I want to kind of focus on that, and that's, it actually shows the major issues with the gene therapy. So we started research, and that's why in January, 12 years ago, we gave the first money, and the AGU mice was purchased. So it took 5 years between that research, and we had a pre-IND meeting. So this is the meeting with FDA where you show everything that you've done, and you show your proof of concept, you show your like all of the data, the efficacy data that you developed for the gene therapy. And actually that meeting with FDA, that once that was held 5 years after we started the research, was actually a very good meeting. FDA told us what else they needed to do, what else is needed to start the clinical trial. That meeting with FDA is actually, from this time to the time we started clinical trial, saved us about $2 million, just to let you know. Because during that meeting, we were also asked and was approved of what type of animals to use in the toxicology study, which is, it's over here, is called GLP study, but it is toxicology and safety study in animals. And we needed to use like animals that are cheaper, because as default, you have to use non-human primates to test that. But because the experiments, go ahead. But the experiments was designed pretty well, so FDA liked everything. Then you can see that there was a pretty big time between the 2018 to 21 when we started doing the production. So it was 3 years. That was actually the time when the pharmaceutical company came over and started doing the work. But eventually realized that, but it took them about a year to do the assessment. Then another year, they start gathering the team and putting everybody together, start some interviewing some patients, doing patient assessment. And then another year, it took them to drop everything. Because in the end, they realized that it's really non-profitable for them. So that was the waste of kind of time in for our development, but it was a very good learning experience for us. The drug production, the drug, and I mean the gene therapy drug, it takes about a year to year and a half to produce. It's just who is helping with the testing? Sorry, which type of testing? There's lots of testing was done here.
Yeah, that's what we're actually trying to figure out now, is who we can work with to help with the different experiments along the way. Some will be Curtis related, some Curtis can't do, some will have to be, like Julia said, primate studies that we have to eventually downstream work with. So I am in active discussion and trying to find teams that do this kind of work, but
yeah, well, I mean, we can talk more detailedly about that.
Yeah.
Like if you want. So, and okay, we talked about that one, and this is how much it costs so far. This is just for you approximately. So everything that's before pre-IND is probably about $500,000, and after that it's somewhere around $5 million. And
so we're just
majority,
we're going to sell a lot of t-shirts, guys. Because Julia
well,
used this money somehow as well with their community.
so like one of the tricks is, yes, that's what I was telling. So the GLP study, if you're doing it in non-human primates, it's about $2 million, probably $1.5 million to $2 million. But the FDA gave us in the pre-IND meeting go ahead to do rats as a large animals. So for us, it was only 500, like $500,000, which you see, you can immediately take out 1.5 million out of that 5. So there is lots of other little things. So it's not that like you need to be kind of scared of that 5 million. It's just all about, continuously connecting. Like there is a few people I talk to. I mean, there is a really good foundations who want to do that, but they don't go to conferences and they don't go to, let's say, to networking events. And that's where all of these things happened, actually. That's majority of the money that I was able to negotiate down out of those 5 million is actually trying to kind of piece it together by talking to people, by connecting to people and things like that. So one little step at a time. All right, and that's kind of the slide that was before. It's really kind of the roadmap from the proof of concept to clinic. So, and this is not, sorry, went to a little bit too far. This is not, this one, the picture is not my slide, so kind of the general slide, understand the disease mechanism. And I think you already know the disease mechanism, but what I wanted to keep it here, the reason why I want to keep that picture here is because it's very important once you are developing the gene therapy, for example, if you are planning, or any kind of other type of the genetic therapy, test it in fibroblasts. Do all of fibroblast testing. It will actually save you tons of headaches in the future, and can actually have a lot of benefits with that. The model characterizations, you probably have already the mouse model for that, correct? Yeah, so it's very good. So the gene therapy development, when I started 12 years ago, it was still kind of very how you would say that, probably very boutique approach at that time. Right now, it's almost the standard approach. The development of the gene therapy can be done by high school student almost. It's a software now you do that, and it's not that difficult.
And we're actually going to have Philip come back and talk to us. He's one of the researchers that works on PDE with Curtis, who's on the call, and he's gonna talk to us, I think, about a gene therapy that they worked on for a different rare disorder. So we have some folks in our research community who are kind of are knowledgeable of in that space.
Yeah, that's great. So then you conducted safety, the efficacy and the dosing studies in a mice, the ones that you have already. This is the time frame when you're going to do that pre-IND meeting, because then you would need to decide, well, it's not you need to decide, it's the FDA needs to give you the advice, what you can use for your safety studies and tolerability studies. And once it's done and the drug is produced, you're going to the clinical trial. So this is the questions you would need to answer when you will bring your product to the FDA and to the clinical trials, to the PI, to adopt them. It's first, whether it can be delivered to where you need it to go. So you would need to understand actually where it needs to go. There are lots of different routes of administration, and in those efficacy studies, you need to conduct them the best way. You need to be able to translate into humans. Then the efficacy, whether it will do whatever you want it to do to make patients live longer life, better life, and more functional life. And whether it is safe, whether is this going to harm patients or not. So with this is the questions you need to answer before going into the clinical trial. And once you answer all of those questions, that's FDA is making the assessment whether that is enough benefit versus the risk of the patients. So that's kind of goal of those assessments. The reason why I'm pointed to to you is the community, because there is a lots of work that we were doing, and lots of work I can see lots of other people are doing, is actually trying to learn more. There is lots of groups are doing a lot more work than those three. This is what you need to do to treat the patient. You don't need to kind of do a lot more research and a lot more people are overdoing it.
Yeah, they're getting information not relevant to the most direct
Yeah.
path to therapy, right? So if you're trying
Yeah.
to follow the most direct path to therapy, then answer these 3 questions and carry on.
Yeah. Not sure if so this is one of the things, is what is the drug it's all about. The drug for gene therapy, and one of the things that what I was talking is, so the steps of in our human body, what we have in DNA is we have something that's our DNA is made out of the little pieces, the introns and exons, and eventually the exons are the parts that is translating into the protein, and introns are all cut out and left over. Most of the time, they're used just to keep the stability of the exons in place. And that's what the drug, what I was talking about, the drug for the gene therapy, what it's building, you literally taking that particular blue piece, and this is the blue piece, that's your coding sequence, and that's why it's called transgene, because that particular gene is directly translated into those first amino acid chain and then the protein. And you are putting there, or you're left with the promoter. This is the promoter. That's promoter is something that starts, and you add in like one little intron just for the stability for that whole piece. And this is the whole sequence. That's what's called your drug. So literally you are replacing that gene that is person is either missing or non-functioning in the body, and put it back in the body. That's what is called that's what is cost the most in the drug production, is that's what costs the most out of that 5 million. So it is somewhere dependent on how much do you need. It's cost somewhere between to start clinical trials, it will cost you probably close to between $1.5 million to 3 or 4 million. So essentially, you literally, the point of this slide is you're replicating what's is in the body and putting it in. the different types of drugs that are developing is you're going to change different promoters. You can take human promoter, but sometimes there is a better promoter that can be used. You change in different promoters. There is a variation of the code and sequence that can be done that will result in the same protein, but just a variation of code and sequence that will help produce, sometimes produce stronger or better protein. And that's kind of it. So that's, but as a result of the protein that is needed will be exactly the same protein. This is something that I kind of put together based on the work that I've done. What I think the development steps should be for developing the gene therapy, what key takeaways, what you need to look at, and you will have all of that. I'll give you the copy of that, and what really needed to be done step by step, and how much how much it can cost. So that's, I know that's like, it's too much information right now, so we can talk more about those
I guess
details.
the point I want people to take away is that there is a road paved. Yes, we have to follow it. Yes, it's not easy. But there is a road and there is a way. It's like Oregon Trail to me. I'm reading those books again with my kids. And you have to make smart choices along the way, but there is a way to get to the Oregon territories, I guess. Yeah. And I think that's reassuring to know. I guess that's why I appreciate you so much, is it's reassuring to know that there's folks doing this. And I'm not saying that we have to do this as a community, but if this is a road we choose to take, or a part of this road that we choose to take, you know, there is a way that we could do it. So, and also things have advanced since 2013, right? If you were starting today, things would be very different than if you were starting this in 2013 when you started. Yeah.
Yes.
Can we take questions, Julia? Is that?
Yeah, that's it. I mean, it's only the pictures of our kids, different our community kids.
Oh, that's cute. Do you guys have any questions from folks? Because it's really, the goal is for us to try to extract as much sort of understanding as possible. Anybody have a question?
I would have a question.
Sure.
So I assume that something got done in the 12 years. Isn't that what you're saying?
Oh yeah, I mean, we're about to start clinical trial. We did all of that.
So, I mean, did your sons get it?
Hopefully this year. We produced the drug. We did all of the, I mean, right now I'm still in the process, but I'm about to submit IND to FDA to start clinical trial. So we have the doctors already lined up, everything. So my goal is to have treated first patient by summer.
So how did you manage to get all that money in such a short period of time, especially when you were probably like the first people to do it?
Fundraising. It's so the goal for fundraising, yes, you really would have, you tell people the big goal, but then you break everything by chunks. Yes, you break everything, like you say that, okay, we are doing the cells, we're trying to test the drug, and for this we need $10,000. $10,000 is not much. So literally you can between your community, you have probably like 10 families here or more, 15.
Yeah.
So 15 families. I would say you probably need not more than $500,000 to get to pre-IND. So with 20 families, it is what, 25,000 fundraising in 3 years. I think you can do that.
So I think if I can, I think what's really cool, and
Divide and conquer. That's my
right.
answer.
When this all first, like for me, when my adventure with all this first started, my only goal in life was to get it on the newborn screening card. In the last year, now the goal is gene therapy. I never even thought that was it never once occurred to me. So I think what you said with the fundraising and building relationships and things like that is showing people that there's more to it, right? Like, and just telling the whole story and then the many goals, right? So I only ever talked about the newborn screening, but now like my story's changed, like that's just a block in the road to long-term cures, if you will, for PDE. So I just want to say thank you, because I realize it's a long road, right? But I think people like you who essentially discovered it and figured it out and built this along the way as a parent, I think is very inspiring. So thank you for that.
Thanks. Yeah, I mean, I haven't started the newborn screening journey yet, so I have a lot to learn from you. So,
I met a mom who's very active with the SMA community, and her son was one of the first 4 kids who was diagnosed through newborn screening and received the gene therapy for SMA. So their gene therapy is a little bit different. I believe it was an ASO, so it's an RNA-based gene therapy. But just speaking, I guess, like, I would love a future where kids are diagnosed and receive therapy before they're even symptomatic, whether with this gene therapy, whatever, in any way. I guess that's the future. So the two obviously go very much hand in hand, because if her kiddo was never diagnosed with newborn screening, it wouldn't have turned out that way. So yeah, Curtis, I think the one that she was given was ASO, but I don't know specifically. But yeah, there's a lot approved. And to that point, I guess, like, now I accidentally got myself like deep into the SMA community. And a lot of their discussions are basically arguments with pharma about who owns which of their gene therapies. I think if I remember correctly, there's something like 7 therapies that have been developed for SMA, but they're all under the control of one or another pharma company. And so I guess that's what appealed to me about Julia and her path, is that no one else owns this, except her.
so one of the things that what you need to, and that's probably is about, there is lots of legal things that you need to put in place once you start in that. So one of the reasons why I said that that gene therapy drugs can be developed by high school kids. Essentially, what I like from what I see that lately I've seen the approaches is let's step back. You're completely correct about that fighting about the IP. Well, IP is intellectual property, about fighting on IP for the ultra rare diseases. That happens so much in that ultra rare disease community. You probably can see that there is a company in Dallas that picked up a lot of ultra rare disease. I think at one point they had like almost 20 indications. And I think lots of parents had to sue them to be able, because once somebody owns commercial IP, there's nothing you can do. You cannot go back and you cannot do any research and stuff like that. What I did when I started that, and that's how kind of I was able to pick up, because the pharmaceutical company that was doing our development, they got the IP from university that developed our original product. But when I signed that contract with university, first of all, I signed the contract. So never and ever, and Anna knows probably, give a donation to university, give a sponsorship, sign a sponsorship agreement. So what it gave me, that with sponsorship agreement, I can freely do, so I have the IP for research and development, and educational rights, that's what it is. So everything that up to the clinical trial, up to the end of the clinical trial, it is considered to be a research. So I always can do everything without that. And then eventually we picked up the commercial rights too. But when you're developing things, and that's one of the pathways that can be done now, there's lots of CDMOs, and CDMOs is the company that producing the drug. There's lots of CDMOs, or the company that's doing the gene sequencing, they actually can produce that drug for you. There's tons of companies that's done, but the software is always the same. But what it will do for you versus going through university route, then you will own that IP, and then you can bring that drug and tell university to do all the rest of the research. But then university will not be able to negotiate the rights to your development. So essentially, then the kids will be safe from all of those legal implications and legal fighting.
Yeah, there's a lot to consider. I actually have to drop, guys. I have a 2 o'clock for my regular meeting. Are there any questions, or can we all disband for today? If you guys have other questions, just email them to me and I'll pass them along to Julie. And this is a conversation that we're obviously going to be having for a long, long time, for many webinars forward. And if you guys ever want to get together and chat outside of the webinars, I'm also happy to do that. I'm going to stop the recording.
I can also stay